biology. What we're talking about right now is going to be regulation of gene expression. So we talked in the last chapter about how those genes are actually expressed, and now we're going to be talking about how we regulate which genes are expressed and which ones aren't. Okay, so first of all, I mean, there is such thing as differential expression of these genes. We're going to express different genes depending on our surroundings, depending on our environment. So in prokaryotes, they're going to be regulating gene expression in response to environmental conditions. And then in multicellular eukaryotes like us, it's going to regulate development and also differences in cells, and then also responses to our environment. And RNA molecules are going to play a bit of a role in regulating the gene expression as well. So this is an example of a fish, and this fish has differentiated gene expression in the top half of its eye versus the bottom half of its eye, which allows it to see in the air with the top half and in the water with the bottom half, and it can see equally well in the top half and the bottom half of its eye in air and water, which is kind of crazy, but it's just an adaptation that this fish has. All right, so let's talk a little bit about regulating the transcription of the genes themselves. So bacteria do better that only produce the things that they actually need. So you can probably imagine how it would be advantageous to not be producing a bunch of things that you don't need all the time. And so we can regulate, or bacteria is what we're going to be talking about, can regulate the production of enzymes by what's called feedback inhibition or by gene regulation. We talked about feedback inhibition earlier. And so one of those methods that they use is referred to as the operon model, and we'll talk about what operons are in just a second. So here's feedback inhibition. We talked about this before. So you have a precursor to tryptophan here, and then you have 3 different enzymes that are involved in making that tryptophan. Well, once that tryptophan's made, once there's enough of it, that can go back and act as an inhibitor to this enzyme. So that's one way that you can kind of regulate things. Another way is that tryptophan also can work in the regulation of gene expression, and we'll see how that happens. We'll look more at that example in just a second. So if you have a bunch of genes that are closely related, like the genes that would encode for these 3 enzymes, those can be controlled kind of all together by a type of switch, which is referred to as an operator. So that's a segment of DNA that can be, essentially turn off the expression of those genes. And operon is the word that we refer to, is the word that we use to refer to the operator, as well as the entire promoter region and the actual genes that are controlled by that operator. And this might sound a little bit crazy, but we'll see some pictures. Or if you have your books out right now, you might be looking at some pictures and it might make a little bit more sense. So that operon can then be switched off by a protein called a repressor. So the repressor is essentially just going to bind to that operator, and it's going to just physically block RNA polymerase from creating RNA, from transcribing those genes. And so it's really just that whatever that repressor is binding there, and it's preventing RNA polymerase from transcribing the gene. And that repressor is encoded for by a different gene, which is called the regulatory gene. So that repressor can either be active or inactive as kind of its default mode, depending on what molecules are around. So there are what's called co-repressors, which are molecules that bind to that repressor and they kind of act with the repressor to turn an operon on or off, or to turn it off, because the repressors only turn off operons. So with E. coli, we talked about tryptophan. We're going to look at that example again. So E. coli can synthesize tryptophan if it needs to, but it doesn't want to do that all the time if there's tryptophan around. And tryptophan, by the way, is an amino acid, so it needs it to make proteins. So you have this TRP operon, or we'll just call it the TERP operon. So this TERP operon essentially is going to turn on or off the genes that are necessary to make those proteins that can then make tryptophan. So when tryptophan is around, when there's plenty of tryptophan and the cell doesn't need to make its own, then it's going to turn the operon off. So the repressor is active only when there is tryptophan around, and that tryptophan acts as a co-repressor. So when tryptophan is around, then you don't need to be making your own tryptophan, so you're just going to turn off that whole operon, stop making all of those enzymes that you need to make tryptophan, and just use the tryptophan that's around. Okay, so then the TRP operon, the TERP operon, is then repressed. So this is what this all looks like. So this right here would be the operator. The operon is promoter, operator, and then all of those genes. This is a molecule of RNA polymerase, and so here you have the repressor, and right now it's in its inactive state. Okay? So this would be when there's no tryptophan around. There's no tryptophan around to bind with this, so it's in its inactive state. So this operon is on, and it's producing all of these subunits that are going to make those enzymes that then make tryptophan. Well, when tryptophan is around, it's going to bind to this co-repressor, or to the repressor, because it is the co-repressor, and now you have an active repressor. And now this repressor can bind to that operator, and now RNA polymerase is just going to bump into that repressor, and it's not going to be able to transcribe that gene, those genes. Okay, so that's one type of what we refer to as negative gene regulation. That's one type of negative gene regulation. And that one is usually on, but it needs a co-repressor to turn it off. And so that's what we refer to as a repressible operon. And an inducible operon is one that is normally off, and so that repressor is normally bound to it, and then something needs to come in and turn it on. Okay, so then you have a molecule that's called an inducer that's going to inactivate the repressor. So in the case of the TERP operon, we saw that there was a molecule, tryptophan, that was activating, right? It was activating that repressor. And in this case, we're going to see that you have another molecule called an inducer that's going to inactivate the repressor. Okay, so one example of that is the LAC operon, which makes enzymes that help us break down by itself. That LAC repressor is just going to be active. It's always active by itself, right? The TERP repressor was inactive by itself. This one is active by itself. And it's going to switch that LAC operon off. It's going to be bound because it's active. So then when that operon needs to be turned on, an inducer is going to then inactivate the repressor, and that's going to turn the LAC operon on. So this is what that looks like. Normally, you have the repressor is going to be active and bound to the operator, and that's just what normally happens. And then when allolactose comes in, it's going to bind to the repressor and make it so they can no longer bind to the operator, And now RNA polymerase can activate these genes. So, this is a zoomed in version of what that looks like. These inducible enzymes are usually going to function in catabolic pathways, and repressible enzymes are usually going to be in anabolic pathways. And if you remember from earlier, catabolic, does that use energy or does that produce energy? It produces energy. And anabolic pathways use energy. Okay. Yeah, and so the repressible enzymes like TERP are going to be ones where you're synthesizing something and then the end product is turning off those genes. Okay, and so both of these are negative control. They involve turning those genes off, okay, by the active form of the repressor. So when the repressor is active, it's turning the genes off. That makes these negative controls. And there is also positive gene regulation. So there's also positive gene regulation. And one example of this is a protein called the catabolic activator protein, which we'll just refer to as CAP, and that activates transcription, okay? So then when glucose is around, which is what E. coli prefers to eat instead of lactose, when glucose is not around, then CAP is activated, it binds with cyclic AMP, that AMP stands for adenosine monophosphate, so that looks a lot like ATP, except for it's only got one phosphate and it's cyclic. And then once that's activated, that's going to attach to the promoter region. So remember, it was promoter, operator, genes. It's going to attach to the promoter region of that lac operon that we were just looking at. And it's going to accelerate the transcription. It's going to make transcription happen faster. And then once the glucose levels increase, which glucose is what they like to eat, then CAP is going to detach from the lac operon, and then it's going to return to its normal rate. And let's look at what that looks like. All right, so when cyclic AMP comes and binds to CAP, then in CAP's active form, it's going to go to the promoter region, and it's going to make that transcription happen faster. So this is when there is low glucose and when lactose is around. Because remember that this would be, if there was no lactose around, then this would be active and it would be attached there, blocking the RNA polymerase. All right. So this is when lactose is present and glucose is present. This is lactose present, no glucose around. So when there's lactose present but there's glucose present too, then that's not going to be active. It's not going to be promoting because E. coli prefers to eat glucose. All right. And so that's the end of 18.1. I'm going to stop for a little bit and we'll get back to this in just a little bit.